Meiosis, replication of DNA, chromosome duplication, followed by 2 rounds of cell division, result in 4 haploid daughter cell gametes that possess half the amount of DNA of the parent cell. Mutations also arise during meiosis, crossing over and random assortment occurring during meiosis lead to new combination of DNA. Meiosis. Here the parent cell has 2N diploid. During meiosis we have 4 gametes, 4 sex cells, ova or sperms, has haploid cells. Mitosis occur in haploid and diploid somatic cells. Continuous process, 4 cytological distinct stages. Prophase, chromosome shorten, thicken, and become visible by light microscope. In prophase, centrioles move apart and the mitotic spindle begin to form. Centriole migrate to opposite side of nucleus and nuclear envelope begin to disappear. Metaphase, the nuclear envelope disappear completely. Replicate chromosome held together at the centromere are aligned on equator of the spindle. Metaphase plate. Anaphase, centromere split and the daughter chromosome migrate to the opposite poles. Cell division, cytokinesis begins. Telophase, the nuclear envelope before chromosome become extend and this visible and cell division continues. Meiosis is a special type of cell division that occur in sexually reproducing organisms, producing the gametes, sperm or cell egg cells that contain half the number of chromosomes as the original parent cells. It involves 2 rounds of division called meiosis 1 and meiosis 2, with DNA replication proceeding both rounds. During meiosis 1, homologous chromosomes joins as tetrads and undergo genetic recombination, exchanging genetic information and forming chiasms between them. The cohesion between sister chromatids is released during meiosis 2 and they segregate from one another to produce 4 haploid cells, each with only 1 copy of each chromosomes. The haploid cell produced by meiosis from a male and female will fuse during fertilization to create a cell with 2 copies of each chromosome, again the zygote. Here the detailed intraphase mitosis in the animal cells. Early prophase, middle prophase, and metaphase, early anaphase, late anaphase, and here telophase, and here the nucleus. Meiosis, the germ cell, occur at a particular point in the life cycle. 2 successive division, meiosis 1 and meiosis 2, of the diploid 2N nucleus after 1 DNA replication. Cell division, cytokinesis, result in 4 haploid 1 cell from single parent cell. In the animal, in gametogenesis, we get gametes. In plants, we call it sporogenesis, and we get meiospores. In meiosis 1, the detailed phases, chromosomes are reduced from diploid to haploid, from 2N to 1N, and 4 stages. Prophase, similar to prophase of mitosis, except that homologous chromosomes pairs and crossover spindle apparatus begin to form and nuclear envelope disappear. Metaphase, chromosome pair bivalent align across equator and random equatorial plane, and random assortment of maternal paternal homologous occur, different from the metaphase of mitosis. Anaphase, homologous chromosome pairs separate and migrate toward opposite poles. Chromosomes in telophase, chromosomes completely migrate and the new nuclear envelope form, followed by cell division. In meiosis 2, similar to mitotic division, also 4 stages. Prophase 2, chromosome condense. In metaphase 2, spindle form and centromere align on equatorial plane. In anaphase 2, centromere split and chromatid are pulled to opposite pole of the spindle. And telophase 2, chromatid complete migrate and the nuclear envelope forms and cell divide, resulting in 4 haploid cell. Each progeny cell has 1 chromosome from each homologous pairs, but these are not exact copies due to crossing over. Here the detailed intraphase and mitosis in animal cell, as we mentioned. Here in the stages of meiosis in the animal cell, the exception is the crossing over and the random assortment. Here gamete 4 getting 4 cells from crossing over and random assortment. There is a difference from the parent cell. Comparison of the mitosis and meiosis in diploid cells. Here the mitosis, DNA replication. In prophase, duplicate chromosome line up individually on spindle. In meiosis, a pairing of homologous chromosomes and duplication become visible. Homologous pair of duplicated chromosome tetrade line up on the spindle. Here occur crossing over and random assortment. Comparison, we here get 2 cells, we get here 4 cells, and as we mentioned here in the 6 cells, the ova or the sperm, twin chromatoid chromosomes and cell divide. In metaphase, anaphase, telophase, we get 4 cells. Here we get 2 cells. Significant result of meiosis: haploid cells are produced because 2 rounds of division follow 1 round of chromosome replication. Alignment of parental and maternal inherited chromosome is random in metaphase 1, resulting in random combination of chromosome in each gamete. Number of possible chromosome arrangement 2 raised to 2 minus 1. Crossing over between maternal and the paternal chromatid during meiosis 1 provides still more variation. The crossing over site vary from meiosis to another. Here we spermatogenesis and oogenesis. Here the parent cell, primary spermatocytes, secondary spermatocytes, and here the spermatid in the testicle, a spermatozoa, meiotic division and differentiation. In oogenesis, oogonium has 2 N, primary oocyte, 2 N, first polar body and secondary oocyst, oocyte, and secondary polar body.